Hello, it's Mittali from Group 319 and here I am presenting my topic thermodynamics of melting of the double helix. So we started thermodynamics of melting of the double helix. The melting of the double helix, particularly in DNA, refers to the process of denaturation where the two strands of the helix separates. There is how temperature affects the stability of a double-stranded structure. The melting temperature is the point at which half of the DNA strands are in the double helix state and half are in the melted single-stranded state. There are various key factors influencing the melting of double helix include. So we will discuss about the various key factors that influencing the melting of the double helix. Now first one is base pair composition. In the base pair composition, the presence of GC pairs which form 3 hydrogen bonds contributes to a higher melting temperature compared to 80 pairs which have only 2 hydrogen strands. GC and 80 are DNA base pairs which are we will study in this chapter. Next one is salt concentration. In the salt concentration, high salt concentration stabilize the double helix by shielding the negative charges on the phosphate backbone. These increase the melting temperature. Now we will start the third one. Third one is length of the DNA strand. In the length of the DNA strand, longer strands units they have higher melting temperature due to the increased number of hydrogen bond and stacking interactions. The process can be described using the Gibbs free energy equation, where the change in free energy determines the favorability of the melting process at the melting temperature is 0, indicating a balance between the double-stranded and single-stranded process. The process can be described using the Gibbs free energy equation, where the change in free energy determines the favorability of the melting process at the melting temperature is 0, indicating a balance between the double-stranded and single-stranded forms. This process we will discuss in 3 or 4 types like melting temperature, enthalpy, entropy, Gibbs free energy. So first we discovered about melting temperature. Melting temperature, this is the temperature at which half of the DNA strands are in the double helix form and half in a single stranded state. Melting temperature depends on factors like base composition and strands length. Now we will discuss about enthalpy. The melting process involved breaking hydrogen bonds between base pairs. The enthalpy for melting is typically positive, indicating that heat is absorbed during the transition from double-stranded to single-stranded DNA. Third one is entropy. Now we will discuss about entropy. The transition from a double helix to single strands, less order state, and double helix is more order state, increase entropy. This entropy is also give positive during melting. Entropy and the transition from a double helix is a more order state to single strand is less order state, so increase entropy. Thus entropy is also give positive during melting. Fourth one is Gibbs free energy. The relationship between enthalpy, entropy and temperature is given by Gibbs free energy. Now we will discuss about pH and environmental conditions that affect the passive transport. Interim pH level or other environmental factor can affect hydrogen bonding and base stacking interactions, influencing melting behaviors. The double helix is the structure of DNA consisting of two intertwined strands that resemble a twisted ladder. Each strand is made up of a sequence of nucleotides, which include a sugar, a phosphate group, and a nitrogenous base. The base pair is especially adenine with thymine and cytosine with guanine. The base pair especially are adenine with thymine and cytosine with guanine. Holding the strands together, this structure is crucial for DNA replicates and the encoding of genetic information. It was famously discovered by James Watson and Francis Rich in 1953. Thank you.